right, everyone. Thank you so much for being here. My name is Dion Rossiter. I am the Executive Director of Science at Cal. It is so great to have you here. I am joined by Rosalind Sarvey, also at Science at Cal. Hi, Rosalind.
Hi, Dee. Hi, everyone.
And also Elaine Liu, who is our guest host for the day. She is at within the School of Education. She's a graduate student. So hello, Elaine.
Hi, everyone.
Where is the farthest person tuning in from? Last time we had somebody from Australia. Is there anybody that far away?
No, I don't see anyone from Australia, but we've got folks from Indiana, Florida, North Carolina.
So we got some East Coasters in the house. Okay. Well, it's nice to have you here. If this is your first time joining us, welcome. If you are a return viewer, welcome back. Again, this is our Grounds for Science event. We have a wonderful event today. We have two speakers, both graduate students, one in the Department of Chemical and Biomolecular Engineering. And we have one from the Department of Epidemiology and Biostatistics. That's Anna Carneiro and Navya Pothamsetty. So welcome, ladies. We are so happy to have you. And again, welcome everyone else for being here. Just to start us off on the right footing, we want to, at Science at Cal and UC Berkeley, recognize that Berkeley sits on the Huchun territory, the ancestral and unceded land of the Ohlone people, the successors of the historic and sovereign Verona Band of Alameda County. This land was and continues to be of great importance to the Ohlone tribe and other familial descendants of the Verona Band. Every member of the Berkeley community has and continues to benefit from the use and occupation of this land since the institution's founding in 1868. By offering this land acknowledgement, the Berkeley community not only recognizes the history of the land on which we stand, but also recognizes that the Ohlone people are alive and flourishing members of the Berkeley and broader Bay Area communities today. So thank you for allowing me to take the moment to acknowledge the land that we are on today. Well, some of us are, but all the native lands. Science at Cal, like I said, some of you may be new to Science at Cal. We celebrate science by bringing the wonder and the excitement of UC Berkeley STEM research to the community. All of our events are free and geared toward a public audience. So you came to the right place if you want to learn about Berkeley research from all across the STEM disciplines. We have programs like this one. We have science cafes, traditionally live. So of course, we are coming to the end, hopefully, of shelter in place. And we will meet you back inside our cafes and our restaurants and our coffee shops and our lecture halls soon. But until then, we will be all virtual. You can find us at festivals around the Bay Area and Cal Day on campus. We have partners across the Bay Areas from consulates and libraries, and we do First Friday events with the art community, farmers markets, community centers, you name it, we are involved. If you have an idea or other interesting ways in which Science at Cal can be involved in the community, please let us know, shoot us an email. We want to thank you, of course, for being here, and we want to thank all of our supporters, which includes all the people who donated today. There's lots of different ways that you can be a part of Science at Cal, and one of them is supporting our programs monetarily. And we just want to thank you so much for doing that. We provide stipends for grads. We provide food for some of our underserved communities. We have lots of different ways that you could help us out by sponsoring some Science at Cal programs. And if you want to learn more about how to do that, please also contact me. We have a few programs that are on the docket, so I hope you can join us. I just want to let you know of 2 or 3 that are coming up. One is next week, Adapting to Change, the Future of California's Water Energy Nexus. The next after that is in 2 weeks. This is one on the Oakland Eco Block. So be ready to or stay tuned for those and join us. I hope you'll be able to come and with us. I just wanted to mention I made my drink. It's not the Aperol Spritz though. It's the mocktail option because I didn't want to get sloppy while having to host this event. But I hope some of you This was inspired by Navya, right? Navya, she wanted the Aperol spritz because the weather has been so beautiful, which it has been today also. So I hope some of you are enjoying a nice beverage on behalf of Navya's idea. So thank you for that. We're going to go through some trivia first and foremost, and then we'll go ahead and have Anna give her presentation. After that, we'll have a short Q&A. This is my time to remind you to always add your Q&A questions in the chat box as often as you'd like throughout the entire presentation, but we will be asking Anna just 1 or 2 questions at the end of her presentation. We'll then go into some trivia with Navya, where she has some questions she'd like to ask you all. And then we'll go ahead and go into a short Q&A with Navya. Then we will bring all of us together, bring everyone back, and we will do a Q&A, and we're going to have that open to the whole audience. So if you look at your screen, your Zoom screen, you'll see, I think it's called, let me see, mine looks a little bit different than you. Reactions, is that what it's called? I don't see that. Is that what, reactions? Okay, good. That's just from memories. From my memory, you'll see the reaction button, and if you click the reaction button, you'll see a raised hand function. During the open Q&A, you are allowed to raise your hand and unmute yourself, and you'll have an opportunity to ask the question, any question you want, to either of our speakers, or you can continue to put your questions in the chat, and one of our hosts will answer those questions for you. So again, lots of questions, lots of, this is meant to be like we're in a cafe, so we're going to be drinking mocktails or real cocktails, and we're going to be asking lots of questions and having conversations with our beautiful, brilliant scientists. So with that, I am going to stop sharing my screen, and I'm going to hand it over to Rosalind to introduce Anna.
Thank you, Dee. So before I introduce Anna, we're going to actually go over some of her trivia questions. So I will go ahead and introduce her after that. She'll be actually helping us with the trivia questions. So we'll be posing the questions to you as a poll. You can select your answer, and then Anna will share with us the correct answer. So let's go ahead and dive into it. So our theme for the night is virus trivia. And so the first question is, what was the first virus discovered in humans in the 19th century? Was it yellow fever, smallpox, vaccinia, or the tobacco mosaic virus? So go ahead and put in your answer. And once we've got the answers coming in through, we will go ahead and reveal the correct answer. So I see, kind of looks like people are coming to a consensus, but lots of different answers.
I don't know the answer to this. I didn't look at the trivia questions beforehand, so I'm a little clueless.
Wow. All right. It looks like we've got most of our answers locked in. So let's go ahead and close the poll. All right. So lots of people said smallpox. So Anna, what is the correct answer?
The answer is actually yellow fever. So yellow fever had been causing disease since about the 1600s. And then in the late 1800s, Cuban physician Carlos Finlay discovered that it was an illness transmitted by mosquitoes, but they still didn't know what viruses were. They just thought it was caused by a filtrable agent. And then it wasn't discovered that it was a virus until the 1900s.
Fascinating. I think smallpox, if I'm not mistaken, was the first to have a vaccine. So maybe that's why people were thinking that. Yeah. All right, let's go ahead and go to the second question.
I was going to say, we all learned something new that time, huh? All right. Launch poll.
All right. What percentage of human DNA comes from viruses inserted into our genomes in the distant past? This one may be a little trickier. I feel like people are taking longer to respond.
And there's like almost an even split. We have lots of people voting. That's exciting.
Very exciting. Wow, lots of different answers here.
Ooh, oh, we're almost at 100%. I'm going to go ahead and end the poll.
Awesome. All right, it looks like most people said 2%, followed pretty closely by 8%. Anna, can you share with us the right answer?
Yeah, the answer is 8%. So there are a lot of viruses that exist that when they infect you, they actually insert their viral DNA into our genome. And so from the distant past in our pre-human ancestors, it's actually thought that 8% of our DNA has viral origin.
Wow. All right, moving on to question number 3. This will be our last trivia question on viruses. What virus does not cause disease in humans? Is it the coronavirus, Adeno-Associated Virus, Variolovirus, or the BK virus? And I'm sure Anna can explain maybe a little bit more about what all of those are. All right, I see no one is picking coronavirus, so we're all on the same page
So
there.
we know that for sure.
Context clues. We got it. We got a smart crowd here. All right.
All right, we're almost at time. Let's go ahead and see the results here, some split decisions. Looks like most people said the BK virus, followed by Adeno-Associated Virus. Anna, can you tell us a little bit more?
Yep, the answer is Adeno-Associated Virus, or AAVs. So the other two viruses, Variolivirus is actually the virus that causes smallpox, and then the BK virus is a group of viruses that are known to either be asymptomatic or cause mild respiratory disease.
Oh, very interesting. Well, Anna's presentation is actually going to be focused on Adeno-Associated Virus, so we will learn a lot more about that in just a few minutes. So thanks so much to everyone for participating in our trivia. That was a lot of fun. I certainly learned a lot. So we are now going to tune into our first presentation by Anna. So I'm going to go ahead and introduce her. So Anna Carneiro is a PhD candidate in the Chemical and Biomolecular Department at UC Berkeley. Before coming to Cal, Anna studied chemical engineering at Purdue University, where she developed fluorescent protein sensors to monitor DNA changes in live cells. During that time, she discovered an interest in research with biological applications. She then joined Dr. David Schaefer's group at UC Berkeley for graduate school, studying how viruses can be used to treat diseases. Anna examines how adeno-associated viruses, or AAVs for short, can be modified to act as safe, effective, and targeted delivery vehicles for potential therapeutics. Outside of the lab, she enjoys volunteering with Bay Area scientists inspiring students, or BASIS for short, getting boba, watching football, and going to comedy shows. We are so excited to have her here, and I'm going to let her take it away. Thank you so much, Anna.
Thanks, Rosalind. So as she mentioned, my name is Anna Carneiro. I'm a PhD student in the Chemical and Biomolecular Engineering Department. And today I'm going to be talking about viruses and how we can engineer them for therapeutic development. If I can move my slide forward. Okay, so as humans, we are all made up of cells, as are all living organisms. And inside each of our cells, we have a copy of our own DNA. And our DNA contains the instructions to help our bodies develop, survive, and reproduce. And our DNA can be broken down into smaller subsections called genes. So humans have around 20 to 30,000 different genes. And the main function of our genes is to produce proteins that help our bodies with our day-to-day functions. So for example, you may have a gene that produces a protein that contributes to the green pigment in your eyes if you have green eyes. Another gene may produce a protein that helps with your body's metabolism. So all these proteins help with your body in some way. And of course, looking at how important these proteins are, if anything goes wrong, that can have a lot of negative effects on our body. So for example, if there is a mutation in your gene, this can have a downstream effect and either cause its corresponding protein not to be produced at all, or for the protein to malfunction, so it doesn't carry out its intended function. So for example, in the disease cystic fibrosis in your lungs, this disease is caused when there's a mutation in a gene called the CFTR gene. And when there's a mutation in this gene, a malfunctioning protein is produced instead of the normal one, and this leads to a thick mucus building up in your lungs. So this is an example of a genetic disease. So any mutation in your gene can lead to these kind of diseases. The field of gene therapy, in turn, tries to address this by providing patients with a healthy copy of that defective gene. So if we're looking at cystic fibrosis, what we're trying to do, well, I'll get my laser pointer. What we're trying to do is we'll look at our lung cells, and these have the defective CFTR gene. And what we'll try to do is add a healthy copy of that gene to the cell with the idea that once it has that healthy copy of the gene, gene function will be restored, and ideally you'll be addressing that disease. But a huge question in this field is, how can we actually deliver these genes to the cell? Can we just inject the DNA into our body? How do we actually get it to our cells? And so the reason this is a big question in this field is because in order to get into the cell, our DNA has to get past the cell membrane, which surrounds our cell. And the cell membrane is a barrier to entry into the cell. So you kind of can think of it as a gate. It'll let some things into the cell and it won't let other things in. So when we just add plain DNA to our body, a lot of it won't actually make it into our cells. Some may slip in, but there won't be enough to actually have a therapeutic effect. So what we actually need to do is we need a delivery vehicle in order to deliver these genes into the cell. So you can kind of think of loading your DNA onto the back of a FedEx truck. And once you package your DNA into the delivery vehicle, ideally your DNA will be able to actually enter into the cell and have a therapeutic effect. But of course, we can't actually load DNA into FedEx trucks. That's unreasonable. So another thing we can use as a delivery vehicle is virus. And so as we've all heard of coronavirus, there's a lot of different viruses that exist in our ecosystem. And they all differ in many ways and in their structure. But the general structure of a virus is that they have a protein shell. So we call this a capsid. And inside the capsid, each virus has a copy of the viral genetic material. And so in order to turn the virus into a gene delivery vehicle, we take out the viral genetic material and replace it with our therapeutic DNA. So going back to our cystic fibrosis example, we'll put in the healthy copy of the CFTR gene into our virus. And the reason viruses are a preferable method of gene delivery as opposed to other mechanisms is that one, the virus shell protects DNA from being degraded by the human body. So our bodies have developed defense mechanisms to destroy any foreign DNA to prevent possible disease or harm to the body, which makes sense. But then if we're using it for therapeutic effects, the body will still recognize it and destroy it. So ideally, we don't want to destroy our therapeutic DNA, and the virus shell adds a layer of protection. Second, viruses infect cells more effectively than plain DNA, so they are able to actually help the DNA get into the cell. So this makes it easier for the therapeutic DNA to make it inside the cell. And kind of related to that, viruses deliver higher levels of DNA to cells. So they're able to get more DNA into your cells rather than other mechanisms. And so therefore we can actually use lower dosages. And this is particularly useful in the terms of safety. Ideally, you would want to administer as little virus as possible to avoid any side effects. And so the virus in particular that I focus on and that I mentioned in the other trivia question is Adeno-Associated Virus, or AAV. So ideally, if we're using viruses for gene delivery vehicles, we would want to pick one that is relatively safe, won't cause disease. So you wouldn't want to use coronavirus as a gene delivery vehicle. So the one we use is AAV, and this is a relatively small virus. It's 22 nanometers in diameter. And just for comparison, coronavirus is about 100 nanometers, so it's about 1/5 of the size of that virus. And the reason why we use AAVs and why a lot of people use AAVs around the world, one is because it does not cause disease in humans. It's not been shown to cause any kind of disease in humans. And second, and really interestingly, it cannot replicate in living organisms by itself. So if you're infected with AAV, one, you won't get sick. And two, the AAV in your body can't replicate unless you're infected at the same time with another virus called Adenovirus. So it's really unique in that aspect and makes it even safer to use. And finally, AAVs infect a wide range of cells. This is particularly useful because there exist a lot of genetic diseases all over the body. So the fact that AAVs can get to different areas of the body make it useful to treat many types of genetic diseases. Of course, there are a few drawbacks to using AAVs that occur in nature. So there isn't just one type of AAV that exists, there's actually several types. And they're very conveniently named. So they're called AAV1, AAV2, all the way up to AAV13. Those are the AAVs that have been discovered in nature. Here I'm only showing the top 9. But the issue with using these naturally existing AAVs for gene delivery vehicles is that 60% of adults have already been exposed to AAVs. So our bodies already know how to neutralize these AAVs. And so this is a problem because if we're delivering therapeutic cargo within our AAVs, our bodies will destroy the AAV before it actually gets into our cells. So there won't be any therapeutic effect. And then 2, naturally occurring AAVs don't exhibit cell specificity. So I mentioned before that an advantage of using AAVs is that they can get into multiple areas in the body. But if we're trying to treat a specific disease, so if we're trying to focus on the lung, ideally we don't want our therapeutics going to other areas of the body, just to minimize potential side effects. So to address this, what we want to do is we want to modify the AAV in a way that 1, our bodies don't already recognize it and won't neutralize it, and 2, may have potentially new properties such as cell specificity. And how we do that is by mutating or modifying the AAV itself. So there are a variety of ways to mutate the AAV, but I'm going to describe 2, for example. So the first method is called Capsid Shuffling, which I'm showing here. And basically what we do here is we take the naturally occurring AAVs, we break them up into small pieces, and then reassemble them all together to form this mosaic-looking shell. So here, for example, you can see this shell that has a little bit of the green AAV, the pink AAV, the blue AAV mixed in. This is all mixed in a very random manner. And then the second type of mutation that I'm showing here is with point mutations. So you'll take your naturally occurring AAVs and randomly mutate them at random points throughout your capsid. So by the end of these mutation methods, ideally what we'll have is we'll have this pool of all these modified AAV variants. And these can range from like 1 million to tens of millions of different variants. Some will probably not work. They won't be functional viruses anymore. But the hope is that some others will have potentially new properties that we can exploit. So with this large diversified pool that we have, how do we find the best AAV variant for our purpose? And to do this, we use the process of directed evolution. And it's called directed evolution because it mimics natural evolution. So going back to our cystic fibrosis example, if we want to find an AAV variant that is really good at targeting cells within the lung, what we would do is we would take our mutated virus library of millions of variants, we would inject it into the lung, and then harvest only the viruses that made it into the lung cells. So ideally, this will narrow our pool down a little bit. We'll then take the enriched library virus and then re-inject it into the lung, and again, only harvest the virus that make it into the lungs. So we'll repeat this multiple rounds, and ideally after each round, our pool will get smaller and smaller and smaller until only 1 or 2 top variants remain. So in this example, we see this purple AAV variant is the one that was the best at infecting cells within the lung. The great thing about directed evolution is it can be applied to different areas of the body. And actually in clinical trials, there are already AAV variants that exist that target different areas of the body, such as the brain, the eye, muscle, liver, and blood. So this is really exciting, and the future of AAV is really promising in therapeutic development. So in summary, viruses are an effective way to deliver healthy genes to various parts of the human body to treat genetic diseases. AAVs in particular are widely used in gene therapy because they are so safe to use and because they are able to effectively deliver therapeutic DNA to cells. We can then engineer these AAVs by randomly mutating them in a variety of ways to discover new enhanced properties that overcome some of the drawbacks of naturally occurring AAVs. And then directed evolution is a process in which we can identify new AAV variants that have the ideal properties we're looking for. So that is the end of my talk. Thank you so much for listening, and I hope you learned a little bit about viruses and AAVs in particular.
Thank you so much, Anna. That was wonderful. I'll wait to see if anyone had any questions. Feel free to put those into the chat. But one question that we had was, if AAVs are so safe, why aren't there more AAV therapies in clinical trials? Could you maybe speak to that?
Yeah, so even though AAVs don't cause any disease, if the patients that want to take these potential therapeutics have organs that are not performing well, it can affect that. So AAVs have a high affinity for the liver. And if someone already has liver damage, if they get a high dosage of AAV, this can affect the liver and have potential effects. And another thing is AAVs, when we're testing out these therapies, we may be testing them in cells in a culture plate, but this is not exactly the same as adding it in humans. So the key species differences can also play a role in that.
Got it. One other question, what are the other types of viruses used in gene therapy? Are there other ones being used?
Yeah, so there are other types of viruses. There are actually a lot of other types of viruses, but in particular, there are viruses called retroviruses or lentiviruses. And these are also very effective gene delivery vehicles, but they are different in that when they inject the therapeutic DNA into the cells, it'll actually integrate randomly into your genome. When AAV delivers its DNA, it exists as a separate DNA location.
Wow, so AAVs are really special then. Wonderful. Well, thank you so much, Anna. We're actually going to go ahead and turn it over to Elaine, who will be introducing the next trivia, but Anna will be back, so feel free to keep asking your questions and we'll have her back later for some more Q&A. So thank you so much, Anna.
Wow, that's amazing. I definitely learned so much about virus in a day, more than ever in my entire life today. So yeah, similar to Anna, Navya also prepared several questions that she's really interested in asking you all as well. So the first hard trivia question we have is, your blood makes a round trip through your body about how many times a day?
This is also a good one. I cannot.
Okay, I'm seeing a normal distribution here. Getting all kinds of answers. We have 74% of the participants have already answered.
This is so interesting to see what people think. All
Yeah.
right, I'll go ahead and end it and share.
Okay, it seems like 52% of our participants think 5,000 times is a good number. What is the answer actually, Navya?
So the answer is about 1,000 times a day. So about 41.6 times per hour, or once every minute and a half. So I guess if it was 5,000 and it was like 5 times, it would be, yeah, a lot. Like it would be going really fast. But I just thought it was interesting that even when we're sitting there doing nothing, like we're still doing a lot.
Yeah, definitely. We have, so at least we have 24% of us got it right. Okay, our next question. Which animal has the largest heart?
Oh, so far consensus. Oh, never mind. Now we're getting different answers. Interesting. I like how many people are voting today. We got a lot of people who, all right, I will go ahead and end and share.
Yes, 76% said blue whale. Sounds like a reasonable answer to me. What is the true answer actually, Navya?
Yeah, so most people were right. Has huge heart of 440 pounds, which is, I can't even think about what that would look like. But yeah, I just thought it was interesting that like there is someone out there with a heart that's like, I don't know, that large. And it's one of those Guinness Book of World Record facts.
Nice.
Awesome. And this would be our last question. Who is the largest funder of health research worldwide?
Elon Musk. Come on, you guys. No one thinks Elon Musk. You can see.
It's pretty close between.
Oh yeah. Yeah, we're I wonder if some people like go in. I don't want to give anyone any ideas. I was gonna say, I wonder if somebody Googles it as soon as they can get their right answer. All right, I'm gonna end the poll.
Okay, US government is the winner here, but is it?
Here.
Yeah, so you're right. And really it's all of, well, assuming all of you pay taxes, like the American taxpayers are the largest funder of health research. So hopefully that makes you a little more interested in what I'm talking about today. But I think that's something that I definitely forget, that it's not like the government, it's like human beings, like American citizens that are paying for research. So yeah, just thought that was a interesting reminder to have.
Yeah, that is a good reminder that money is our money. So that's good.
Awesome. Thank you. Those are great, interesting, very interesting questions. And now please let me introduce Navya. Navya Pothamsetty studies epidemiology and biostatistics in the 4+1 MPH program at UC Berkeley's School of Public Health. She is involved in GRAD's program, the Student Collaborative for Impact Leadership, and is a student representative for the Public Health Alumni Association. In her free time, you can find Navya training her new puppy, Appa, playing tennis, or enjoying the great outdoors. Now, please let us hear from Navya, and please take it away, Navya.
All right, thank you. So today I'll be talking about representation and research, the case of post-traumatic stress disorder and heart disease. And I wanted to start by defining representation in the context of academic research. So when we're asking if a study is representative, we're asking, is our study sample reflective of the population we're trying to make inferences about? For example, if we're testing a new medication, we want to make sure that the people involved in the clinical trial are reflective of the people who are going to use the drug once it hits the market. And as a public health researcher and student, I'm really interested in the differences between individual versus population health risk factors. And the difference kind of lies into the different things that affect individuals versus populations' risk for certain adverse health outcomes. And what I mean by that is, as individuals, we are constantly exposed to things that can make us sick, from the food we eat, to germs, to pollutants, and ideally we have a healthcare professional to take care of us when we have these adverse health outcomes. But there are certain societal factors that make certain communities, not just individuals, more predisposed to having certain health disparities or health or diseases. So when we as public health researchers and scientists look at what can make societies or population sick as opposed to individuals, we look at different risk factors. So just like doctors might ask you about your genetic history or your diet or your exercise habits, we as public health researchers will consider what the Centers for Disease Control and Prevention define as social determinants of health. And these are the conditions in the places that we live, the places that we learn, work, and play that affect a wide range of health outcomes, such as mental health to infectious diseases. And instead of going through and listing a lot of examples of what these social determinants of health might be, I thought it would be more constructive to illustrate how they are put into practice and affect physical health outcomes by an example from here in the Bay Area. So this graphic shows health disparities and social determinants of health mapped onto the BART station map, which is the public transport system that we use in Northern California. And there's a lot going on here, but I wanted to illustrate two key health disparities in this graph. And the first is that there's an 11-year difference in life expectancy between Walnut Creek and 12th Street, Oakland City Center. And the graphic also shows that there's a difference in median household income and a difference in higher education levels between these two communities. And it's important to remember that we're not necessarily saying that if a community has more money or more education, that's the root cause of an increased life expectancy, rather that these factors are correlated, and we should keep them into account as we study how things like life expectancy differ across communities. Similarly, childhood asthma hospitalizations near the Oakland City Center vastly outnumber any of the other ones that is seen on the BART map here. And this is a pretty small geographic area. So when we see a huge health disparity like this, we as public health professionals are like, well, what's going on near the Oakland City Center BART station that's causing so many kids to get transported to the hospital for asthma? And I wanted to introduce this concept of social determinants of health and health disparities and just get you thinking about health on a population level rather than an individual level as I transition over to my research question, which is, what is the association between cardiovascular disease and post-traumatic stress disorder diagnosis among adult civilian populations? And before I get into how this connects to representation and research and social determinants of health, I wanted to give you some motivation behind why I think this is an important question to be asking in the first place. So heart disease, or cardiovascular disease more broadly, is the #1 cause of global mortality, and it's linked to PTSD, which is an anxiety-based disorder. Diagnosis of PTSD, like diagnosis of any clinical condition, is largely dependent on your social determinants of health. So whether or not you have access to a licensed clinician, or whether there's stigma surrounding getting help for mental health, definitely affects your chance of getting diagnosed, in addition to whether or not you actually have PTSD. Finally, I chose to look at civilians because this relationship between PTSD and military veterans was already studied in military populations pretty extensively. And to answer this question, I conducted a systematic literature review. So instead of surveying individuals or looking at individual patients' health data, what I did was ask this question: what research was conducted regarding the association between PTSD and cardiovascular disease, and what are the findings? So you can think of all of the studies that were published regarding PTSD and cardiovascular disease as my little individual units of study. And then I looked at the whole body of literature to see, well, what are people saying and what are the public health implications? And to give you a better idea of what I expected as I embarked on my systematic review journey, was to give you some idea of who's at risk for PTSD. So as I mentioned before, military populations are at risk for PTSD, but so are survivors of natural disasters, immigrant refugees, and survivors of medical emergencies. And these categories don't represent an exhaustive list, nor do they represent mutually exclusive categories, but I just wanted to give you an idea of the wide array of social determinants of health and lived experiences that these populations face that may make them more at risk for PTSD than people in the general population. And what I expected to find when I conducted my systematic review was, okay, so there's going to be studies that I see that look at all these different populations, and I get to sort of see what researchers think the different social determinants of health have to do with these subpopulations' risk for getting PTSD, and maybe some suggestions on how to mitigate that. However, that's not exactly what I ended up finding. So what I found was there were a few at-risk groups that were represented. So World Trade Center survivors, Hurricane Katrina survivors, and Siberian refugees are all uniquely at risk of getting PTSD, and they were represented at the frequencies shown in the parentheses in my study sample. However, these groups on the right were not represented. So no one was looking at PTSD and cardiovascular disease in Indigenous North Americans, refugees resettling in America, and survivors of gender-based violence. Again, this isn't an exhaustive list. I just wanted to give you some comparison between some of the groups that were represented and some of the groups that weren't. And this got me thinking about my findings, because all the groups that I have on the screen here have elevated risks of PTSD compared to the general population, but only the groups on the left were represented. So by excluding the groups on the right, we're excluding a lot of the social determinants of health, a lot of the lived experiences and different factors that contribute to the narrative of the association of PTSD and cardiovascular disease. We're missing a pretty big piece of the puzzle by excluding a lot of different diverse groups and their risks of getting PTSD. And as I kept thinking about this issue, I was thinking, well, why do we see what we see in academic literature to begin with? And this led me to think of 4 guiding questions to help me make sense of what I saw. The first is thinking about who conducts research. So representation in research is largely a function of representation of individual researchers, and thinking about the different backgrounds, lived experiences, and interests of researchers helps us put what we see into context. Second, asking what data are available is also a really important concern when we think about representation. So certain population subgroups such as military veterans and survivors of the World Trade Center disaster have lots of data that's being collected on them with respect to health outcomes. And it's easier for researchers to go and pull from those data than it is for them to collect data on a whole new population, such as say if they wanted to investigate this relationship in survivors of gender-based violence. Thirdly, asking who funds research is really important too. So whether the NIH or a private foundation such as the Gates Foundation determines that a research question or a disease is really important to learn more about, definitely impacts what we see as published research because they're more willing to fund those projects. And finally, asking who publishes research is also a key question because all journals that exist have their own submission requirements and things that they ask researchers to edit in terms of their findings, and at the end of the day have different audiences that they're catering towards, and that definitely impacts what we see as peer-reviewed academic research and also what we see in news articles that quote these papers. And I realized that this was a lot of information about research and just representation in general. And I believe that everyone has a part they can play when it comes to this issue. So I wanted to give you some key takeaways to leave this, this event with. And the first is to ask what's missing. So whether you're reading a paper or you're reading a journalist's interpretation of the paper, just asking what's not there in the study that may make it not generalizable to you, your community, or even to the general population. And this is really important because, as I mentioned before, like we're the ones funding the research, and research is also affecting us because the medical professionals and policymakers that read these articles are making decisions that affect our health and wellness and everyday lives. So it's really important to ask what's missing in these studies and what the studies would look like if they were truly representative. The second is to ask what other people think and to talk about this issue. Hopefully I've convinced you by now that this is a super interesting and relevant topic, and talking about this issue with others will not only help you learn from more diverse perspectives, but also solidify your own understanding about representation and research. And finally, thinking about where you may fit in. And even if you don't conduct research or aren't involved in academia, I think representation is an important norm that we should push to normalize in everything that we do. So whatever your contribution is, just make sure that you are pushing for representation, and that can help us in our journey and efforts to be better and to do better. And thank you. That's all I have today. I hope you enjoyed it.
Awesome. Thank you so much, Navya. And we have some time for just one or two quick questions. There's a question in the chat asking, the 4 at-risk groups you mentioned on the slides, are they the most common or just some examples?
They're just some examples, so they're not the most common.
Okay, and just generally, why are systematic reviews important?
Yeah, so there's a couple of reasons why systematic reviews are important. I think the first is that there's a lot of literature out there, so it's important that researchers from time to time go back and get a bird's eye view of what the consensus is. So like, I guess with context of this topic specifically, like if half the researchers are like, oh, there's no association between PTSD and cardiovascular disease, and half of the researchers say there is an association, then we want to like probe a little bit deeper and see why that might be the case before we have any like policy recommendations or public health recommendations when it comes to a certain issue. Another reason is that we can see if there's any gaps in literature. So as I kind of talked about today, there were a lot of populations that were undercovered, and without doing a systematic review, we wouldn't really have, we wouldn't really know if that was the case.
Got it. Thank you so much. And now we would like to call back Anna and Rosalind so we can have like open conversation. If you have any questions right now, please feel free to raise your hand using the reaction function in Zoom, or you can put your questions in the chat.
Great, thank you all. So we did have a couple of questions come in for Anna. So one that came in a little bit of a while ago is, are AAVs being used currently, or is that something that's just being researched right now, or are they actually being used in clinical trials?
Yeah, so there's quite a few in clinical trials. Right now, there's only 2 that have been FDA approved. One was, I believe in 2017, Luxturna, and so that is known to treat retinal disease. And then there's one that came out more recently that targets another muscular disorder. But right now, there's a huge wave of AAV therapies that are hitting clinical trials, but they just haven't been approved yet.
Great, thank you. And there was one other question, Anna, are adenoviruses common? And if so, why don't we get sick if the adenovirus combines with the AAV that we may be given?
Yeah, so they are a little common, but not super common. So even if you get infected with adenovirus, sometimes the symptoms are like the common cold. So often, if you get infected with adenovirus, you won't necessarily know that. And if you do happen to be infected with AAV at the same time, you still wouldn't be able to tell because AAV doesn't have any known symptoms. And a really interesting fact actually is the Johnson & Johnson vaccine for coronavirus is based on adenovirus. So it does work, and it shows how it can be manipulated for therapeutic processes.
Very cool. I didn't know that about the Johnson & Johnson vaccine. That's a really a cool application of it. Elaine, did we have more?
Yeah, thank you so much. I also did not know about the Johnson & Johnson one. Now I have something I can tell my friends about. Yeah, one question for Navya. What do you think can maybe be done to increase funding for research on groups that aren't really typically represented, like you mentioned in your presentation?
Yeah, that's a really good question. And I think there's a couple different things. So speaking as someone who's doing research, I think doing your best to learn more about perspectives that like aren't really incorporated. So like actually going into communities and like doing work firsthand and like thinking really critically about social determinants of health. And that way you can bring those back to your research. And when you either are doing that work, you're listing certain variables that like maybe aren't really introduced in the research to begin with. Additionally, I think when you're writing grant proposals and you can like insert that like, hey, these certain groups, like what I've noticed in the field work that I've done are that XYZ groups are uniquely at risk for this health issue. So like you should be more aware of them. And that way you're like transmitting your knowledge to other sources of funding. And even if they're not going to like fund your research necessarily, they're still more aware of other groups. And then a third thing is to, again, like talk about the issue. And like, let's say you get funded for a research proposal, but you realize in your work that, hey, there's like these 2 or 3 populations that like aren't covered that should be in your like directions for future research. You can put in like those observations, and hopefully someone will then see your article, see the great research that's being done, and then like they'll take the baton and like, you know, conduct the research that like should be done in other groups. And this is in addition to like having conversations, especially if you're in a position where you can make decisions, or like you're talking to people who can make decisions regarding funding.
Thank you so much, Navya. It looks like we have a hand raised. Adriana or Adriana, I apologize if I said that incorrectly, but if you want to go ahead and unmute yourself, feel free to ask your question.
Yeah, my question was for Anna. Does the Johnson & Johnson virus vaccine have anything to do with AAV that like you only need one dose? Like, is there anything to do with that? Yeah.
So the adenovirus that they're basing the Johnson & Johnson vaccine is completely separate from any AAV. AAV is just named Adeno-Associated Virus because in order to replicate, it needs adenovirus. But the J&J vaccine is just adenovirus. Doesn't have anything to do with AAV.
Okay, and that has nothing to do with it only needing one dose?
No, so because it's a
Okay.
viral vector, it's more efficient and you only need one dose. The Pfizer and Moderna vaccines are mRNA based. So in order for your antibodies and your body to recognize COVID-19, it needs 2 doses in order for you to build up that memory.
Okay, cool. Thank you.
Yeah.
That is fascinating and a really good question. Elaine, did we have any more that you wanted to ask from the chat?
Yeah, we have another question for Navya. Do you know where PTSD ranks in causing of heart disease in the research that you've done?
Yeah, that's a good question. I think so. PTSD hasn't been established as like a cause for heart disease. As I sort of mentioned earlier, it's like you can think about it as a risk factor that's associated with it. So like PTSD won't always be like the reason that someone say gets a heart attack, but it can cause wear and tear on your system through like similar biophysiological pathways that affect the cardiovascular system that will make it more likely that you do get a heart attack. And I think that there's some work being done that's trying to establish like a causal mechanism, but that's kind of hard to do because we can't really do a randomized controlled trial or like control whether or not someone has PTSD and see if that isolated factor is what causes heart disease.
Thank you. That's a great point. Rosalind, do you have other questions?
I do. There was a question in the chat for Anna. We've heard a lot about things like mutating. Is there any concern that a new modification to an AAV might then be able to cause disease in humans? Should we be concerned about that?
Yeah, that's a great question. I don't believe since the base AAV doesn't cause any disease, it would be very unlikely that a mutation would cause it to cause disease, but it is very possible. And that's why we go through the process of trying it in cell cultures first, just to make sure it works. And then we go into animal models to see if it has any poor side effects, it causes disease in those, and then finally human trials. So the whole FDA process is there to make sure it doesn't cause any unintended effects or cause any disease. But it is very possible that some kind of mutation can make it harmful.
That's slightly reassuring. Thank you. One other question that we had was, once the healthy gene enters the cell, how do you make sure that only the healthy gene is expressed and that the malfunctioning gene is no longer expressed?
Yeah. So there are different avenues you can take in gene therapy. So very simply, you can add the healthy gene. But if for some reason the malfunctioning gene is actually producing the wrong version of the protein and you don't want it to be there, instead of putting the healthy copy of the gene in the viral particle, you can actually put in some sort of gene editing machinery. So if you're familiar with CRISPR-Cas9, there are also other methods, but you can actually put that in the viral particle instead. And so when it's delivered to the cells, instead of supplying a healthy copy of the gene, ideally the machinery that you're supplying will actually go and snip up or fix the malfunctioning gene. So there's a lot of things you can do. You can supply the healthy gene, you can fix the healthy gene, or you can try and actually just delete it in the first place.
Very cool, thank you. And CRISPR-Cas9 was actually, discovered and being researched at Berkeley too. So lots of fascinating research happening here right on campus. Thank you. Elaine, did we have any other questions? Looks like some came up in the chat.
Yeah, we have two more questions for Navya actually. How does PTSD directly cause heart disease? Or is it more like indirectly due to any changed behaviors as a result of the PTSD? And this one is kind of touched on in your previous answer, Navya.
Yeah.
Mhm.
So I think in addition, so it hasn't been proved that it's a direct cause of PTSD. Rather, it affects your body system so that your body's more predisposed to heart disease. So that's like one part of it. And then the other part of the story is that post-traumatic stress disorder is actually associated with a lot of risk factors for heart disease, independent of the toll it takes on your body as a disorder. And what I mean by that is people with PTSD have higher rates of substance abuse and factors like that that can affect your susceptibility to getting heart disease. So it's like these two sort of separate ideas that of course aren't totally separate. Like there's definitely some factors that are overlapping there, which is why it's so hard to make about these types of relationships in public health.
Got it. Thank you. Could you also explain what social determinants of health are and how they relate to representation in research?
Yeah, that's a really good question and super important. And I want to thank you for asking that again. Social determinants of health are those that act on community or like societals rather than individual levels. So you can think of, say, like logical determinants of as genetics or your individual like, eating habits or something that's particular to you. And social determinants of health are things that affect your risk of getting a certain disease or have a certain condition or health outcome that are particular of a certain community. So if you remember in the graphic that I showed that there was this one area that was like really susceptible to childhood asthma hospitalizations, like let's say there was like this source of air pollution that was also in that area, like that area a social determinant of health because it's affecting like a community factor is an example of social determinants of health. And how that relates to representation is that different groups based on their lived experiences. So from my presentation, I talked about groups like survivors of domestic violence and survivors of natural disasters and military veterans. And they all are navigating like different social determinants of health that apply to these populations rather than say like, they do apply to individuals, but if we think about how those exposures like affect populations and like studying these group level exposures can kind of give us a better idea of how these risk factors for certain diseases can help us understand what causes these diseases and what are associated with them and how we can better solve them by addressing social determinants of health. So like if we can better prevent cardiovascular disease by decreasing, say like PTSD in military populations, then we can think about interventions maybe like specific to military populations that we can study and implement that'll better help those specific populations because of unique characteristics due to their lived experiences.
Thank you so much, Navya. It does look like there's one more question. Maybe we can make that one the last question of the evening because we are almost at time, but it's such an important topic. So Navya, there's a question, is non-governmental funding doing better than government sources at research fund allocation to ensure better representation?
That's a really good question. I wish I knew because that's now something that I'm wondering as well. And I think that's also, it's hard to see an aggregate too, just because there's some non-governmental funding organizations that have special interests, and they might be better at allocating resources than, say, the government, but there's also branches of the government that are highly specialized. So I think it's important to think about, like, I guess, who's represented in those funding sources too. And to think, yeah, I that's a really good question. And I'll have to think about it.
Definitely. Yeah, a very important question to think about. Well, I would like to go ahead and thank Anna and Navya once again for two very compelling talks this evening. Thank you to all of you for being here. You can go ahead and check out our website at science@cal.berkeley.edu for all of our upcoming events, and you can sign up for our newsletter to check out everything else that we are involved with. So thank you all again for a wonderful evening, lots of really great questions. Thank you to our presenters. And yeah, we'll see you at our next event. So thank you all so much. Bye, everyone. Thank you.